This is the phases of mitosis. Got prophase, metaphase, anaphase, and telophase. Now, between anaphase and telophase, you're also going to have cytokinesis, which we'll talk about later. This is interphase, or late G2. This is actually before the mitosis happened. You've got your molecules, you've got your DNA, which is already duplicated, getting ready to split. You have your nuclear membrane, and you got the centrosomes over here. The DNA has already been copied, so now that's why you see all the doubles, because it's already been duplicated. This is the early prophase. The mitosis spindle over here is starting to form, which will act as a pole later. You also have the chromosomes that are starting to condense, and the nucleus is disappeared. You've got the late prophase, which you'll see is starting to line up. These are the spindles, and you see the chromosomes are starting to attach to the spindles. And the chromosomes at this phase is completely condensed. Now you got the metaphase. You'll see that the chromosomes are now lined in the middle. You have the anaphase, which you see starting to take shape of two cells trying to divide into two. And you see that the spindles are starting to pull the chromosomes towards the pole, which are these. And microtubules starting to push the poles apart. And now you've got nearly the two cells. You got the descending chromosomes, and you got the spindles are starting to disappear. And the membrane is forming again to make a whole cell and the nucleus. So that was the phases of mitosis. Now, looks pretty similar, but a little bit more complicated is the meiosis. Now, this is going to be start talking about the two haploid cells. You got phase of meiosis, which you always start off with the diploid cells, and you are going to end up with the haploid cells. You see that the prophase 1 starting to have starting the cell with the diploid cell, like I said earlier. The chromosomes pair up and exchange the fragments, and you see that the DNA material is starting to cross over, and you're going to have the pairs, the homologous pairs, line up in the middle like we did back at mitosis earlier, starting to split. You got the anaphase 1, and these are called the sister chromatids when they are paired up like this. And then you got non-identical chromatids. So these are a mixture of different DNAs paired up together. And now you end up with two haploid cells. Now, once it becomes two haploid cells, you got the phases of meiosis 2. Now, these stages are going to look very similar to mitosis. You see in prophase 1, they're starting to form the spindles. And you're going to have the spindle growing, and then they're going to be lined up in the middle. You see that the spindle is starting to pull towards the pole, and then you also see the two haploid cells kind of split again. Now you have 4 cells. Now, last but not least, we're going to talk about how the molecules move within the membrane. So you got phospholipids bilayer here, and we are going to say that these are the hydrophobic tails and hydrophobic head. And hydrophobic means that they're basically not loving the water so much. And this basically works like a filter. And it's going to allow some molecules to go through, but not some of the molecules to go through. This is a type of how the molecule can move through the membrane. And this is diffusion. This is the lipid bilayer. It's just a little bit longer. And in diffusion, you're always going to have the molecules try to move from the higher concentration to the lower concentration. You're going to have the carrier protein. We've got the extracellular space and the intracellular space. These are the molecules. And you see that the channels are actually going to catch some of these molecules to be able to go through. And these are very molecule sensitive, so they only pass through a specific or similar molecules. And you got the channels, which is similar to the carrier protein. You got the channel protein right here. And you see that these are allowing go through the channel to allow these molecules or proteins to go through. And just like we talked about earlier, the channels are very specific to different certain type of protein that it's going to allow to go through.